This is a promoter. It binds transcription factors, which recruit RNA polymerases. Where you get promoters, you get expression. It's quite simple, but in synthetic biology, you need to consider your promoter choice very, very carefully because different promoters have vastly different strengths. We're going to consider three different promoters and how strong their promotion is and what they should be used for. So, firstly, we're going to consider the T7 promoter. The T7 promoter is found in the T7 phage for E. coli. Phages don't care about regulation. They don't care about making sure that resources are used properly. They're there to invade the cell and to get the cell to use all its resources on them. So the T7 promoter is one of the strongest promoters that has ever been characterized for E. coli. As a result, you don't want to use it if you're making the cell do something and still want to live, because it will just overwhelm the cell. But you can use it in processes like protein purification. So many protein expression plasmids will use the T7 promoter, as you can have the cells growing, and then right at the moment in late exponential phase, when you want to start inducing protein expression, you can add an inducer, which will find an operator, which leaves the T7 promoter free, and the T7 promoter will just throw loads and loads of protein out of the cell, out into the cell, which is brilliant when you want high yields of proteins. But it's not good for synthetic biology solutions that are organismal, on the organism scale. The next one we'll consider is enzyme levels of expression. So, for example, pyruvate dehydrogenase is an enzyme. Now, these are expressed not so much, well, not nearly as much as T7, and they tend to be under tight regulation. So pyruvate dehydrogenase will only be expressed when the cell actually wants to reduce pyruvate. Enzymes are still expressed quite a lot, though. They're one of the most prominent proteins in the cell because they are metabolic. They're required to run all the cell's metabolic pathways, which are absolutely crucial to cell life. So these are a solid middle ground. And then on our very low end, we have membrane transporters. So, for example, aquaporins, which are found in your kidneys, in your collecting ducts, to let water in and out for osmotic potential, to reuptake your water they use for. Membrane transporters are under very, very weak expression because if you express a membrane transporter too much, it becomes quite non-specific and it lets water and other things just leak out and into the cell how they please. And cells need to be very picky about what they take up and what they let leave, or they just burst because they have way too much osmotic potential. So, these are in increasing levels of expression. But there are obviously a lot of different promoters, which will be for a lot of different processes. And you can find them pretty well optimized in cells for the cell's purposes, because cells know how strong they want things after years of evolution, millennia of evolution. For your purposes, make sure you pick an appropriate enzyme. If you try to express a membrane transporter under the promotion of T7, then your cells are just going to die instantly. Make sure your promoter choice is sensible.